The discovery that DNA is the genetic material began with experiments done by Frederick Griffith, a British microbiologist, in 1928. Griffith had been studying bacterial pneumonia. He had been comparing two distinct strains of Streptococcus pneumonia, one that grew as rough bacterial colonies that did not cause disease. This rough strain was harmless. The other strain of Streptococcus produced shiny, smooth colonies. When these smooth bacteria were injected into mice, severe pneumonia ensued and killed the mice shortly thereafter. As one might guess, when Griffith killed smooth bacteria, the dead bacteria were rendered inactive and lost all their ability to cause disease, and left injected mice happy and healthy. However, Griffith discovered something quite surprising. He prepared a mixture of killed smooth bacteria mixed with live harmless rough ones and used this concoction for the injection of mice. Now, unexpectedly, the injected mice developed pneumonia and died. This was remarkable, as it seemed that some substance from the dead smooth bacteria was able to transform the harmless rough cells into becoming killer bacteria, bacteria that were able to cause lethal pneumonia. What was the transforming substance? This was a total mystery, but certainly it was able to genetically convert the harmless bacteria strain into a disease-causing one. Now, across the Atlantic Ocean, at Rockefeller University in New York, the microbiologist Oswald Avery and his students decided to repeat the Griffith experiment and set out to discover the nature of the transforming substance. Their approach was straightforward. Avery grew cultures of the lethal smooth Streptococcus, broke the cells open, and isolated pure subcellular extracts from them. Each fraction was carefully produced, chemically characterized, and measured for purity. Avery and his team were able to isolate pure proteins as well as pure DNA fractions and tested each for the transformation activity. Each fraction was mixed with live, harmless, rough Streptococcus cultures and then injected into mice. This was to test their effect. Surprisingly, DNA purified from the dead, smooth bacteria was the only fraction able to transform the rough Streptococcus and to convert them to become lethal bacteria. Avery had discovered that DNA, in fact, was the genetic material. So this meant that DNA, a simple polymer of 4 nucleotide letters, G, A, T, and C, must have a structure that somehow fulfills 2 important functions. First, to contain the information stored in our genes, and second, to replicate in such a way so as to ensure flawless inheritance of this information from generation to generation. It was the drive to connect the structure of DNA with these 2 functions that eventually led to the discovery of the DNA double helix. Jim Watson, a zoologist from Indiana, hooked up with Francis Crick, a physicist. Together, they investigated the structure of DNA in Cambridge, England. The 2 young scientists were not the only ones trying to solve the mystery of how DNA worked, but their approach was unique, and ultimately it paid off. They embarked on developing structural models of DNA, models that could satisfy both the functional replication of DNA on the one hand and the flawless inheritance of genetic traits on the other. Their models were based on 2 sets of data provided by other investigators in the field. Most notable was Rosalind Franklin, a chemist working with Maurice Wilkins at King's College in London. Franklin had been applying X-ray crystallography to map atomic structure of DNA. Her exceptional images convinced her and Wilkins that DNA must be helical. The second critical bit of information was provided by Chargaff, who met with Watson and Crick and explained to them Chargaff's rules. Crick reasoned that not only was DNA helical, it was made of 2 helical strands. He proposed that these strands ran in opposite directions. Moreover, it made sense that the sugar phosphate backbones should face outward. Watson, considering the GC and AT one-to-one ratios discovered by Chargaff, proposed that the bases paired and were bound to each other, G to C and A to T, through hydrogen bonds. Watson recalls that on the 28th of February, 1953, it all just seemed to come together.
The Cavendish shop was to build us some tin models, and that took too long. And, finally in desperation, I made some out of cardboard. So here's A, and here's T, and I wanted this hydrogen to point directly at this nitrogen. So I had something like this. Oh, I wanted this nitrogen to point to this one. And I went like this. Whoa, they look the same. And you can put one right on top of the other.
DNA was a double helical structure made of two antiparallel strands, where the nitrogen bases faced inward and paired G to C, A to T. The model was beautiful, and what's more, it just made sense. One strand of DNA complemented the other. The two strands could separate, and each strand would serve as a template in which, through GC and AT base pairing, the complementary strand could be produced, thus ensuring precise replication of our genes.